Thank you very much for the Gardner Foundation and Genome Canada and all the organizers for inviting me to be speaking today. And just because I'm on the organizing committee, it has nothing to do with it. I actually chose to speak, first of all, I should say I've worked 11 years with Eric Lander, and I've so often given the talk just after him. It's just great to have it a little bit of spacer, but then we had Phil, and then we had Svante, so it's always a little tough act to follow after a morning like this. But I'm going to try to talk, go a little further than some of the talks, and particularly Eric's talks, which was talked about cancer genomics briefly, and say how we're thinking we're going to be applying this information in the future. So my talks involved personalized cancer medicine. And I started with a few examples of what you've read in the papers and heard in the news, what personalized medicine, what the promise is going to be for personalized medicine for the patients and the payers and healthcare systems. So we hear about genetic information going to be used to predict risk, optimize screening programs, identify disease at earlier stages, and sometimes say we don't need to give expensive drugs and we can save healthcare costs. The diagnosis will be more precise. There'll be more accurate prognostic information that's going to allow to really optimize the treatment that we give to a patient. And finally, we'll also develop new therapies based on this genomic, these cancer mutations which are identified. And I can tell you, and I'll show you a few examples, that we already have some successes where it's worked, but most of the potential is far from being realized. And some of the difficulties and challenges are going to come through in my talk. A good way to explain personalized medicine is to say right now that there are tests which are being done, which are personalized medicine tests. And there are drugs which are given if you have actually one of those mutations. So the granddaddy of them all is BCR-ABL, is a mutation. We saw this morning a chromosome, a nice karyotype from Svante with 2 chromosome 1, 2 chromosome 2s, and so on. In cancer, that's not the case, and I'll show an example later. The karyotypes of chromosomes are abnormal, so there's particular rearrangement between chromosome 9 and 22, which actually creates a fusion gene, and that fusion gene is a growth factor for patients with CML, a form of leukemia. And if you actually give an inhibitor of that fusion gene, and the BCR-ABL is the name of the fusion gene, if you give an inhibitor, you put those patients in remission. It's not a cure because if people stop taking their drug, the tumor comes back. But it inhibits that very powerful driver mutation in CML, and those patients actually do quite well. Herceptin is a second example where it's a gene that's amplified. An amplification of a piece of DNA makes that there's more copies of that gene. Again, a growth factor. In 15% of women with breast cancer, they have that amplification. And if you actually give a drug called Herceptin to that group, you actually take a poor prognostic group and actually give them they respond to that therapy. At least half of them do respond to Herceptin. But if you don't have that amplification, you don't respond to Herceptin. So it's an example of personalized medicine where you actually did a molecular test to look at a genome abnormality, and you made treatment selection based on the results of that test. I'm going to skip through the others because, 2 years ago we saw BRAF inhibitors in the papers, New York Times, Globe and Mail. ALK inhibitor, I like it at the bottom because it actually shows you that all these targeted therapies are being generated and reach the clinic much faster than before. BCR-ABL took 41 years to actually go from the identification of mutation to actually get approval from FDA. For ALK, it took about 4 years. Only 3% of individuals with lung cancer have that mutation, but the drug which inhibits that 3% of tumors, actually people do respond. Not for a long time, but they do respond. Most of them actually respond for about 9 months. And the reason it was so quick is that you didn't do it the old type of clinical trial where you gave everybody with lung cancer the targeted therapy. You only give it to the 3% of patients which have the mutation. And there's a scientific rationale for that, so we're actually making smaller trials. Those smaller trials are quicker, and with 60 to 80 patients, it's actually possible to get approvals. So I'm going to be talking about different types of cancer mutations. And I'm going to go from risk prediction, which has actually been probably the most difficult, things that hasn't been realized very well yet in terms of genomic medicine, the ability to look at genetic tests and rapidly use that information to predict someone's risk for disease. But I'll still show some examples how things are coming in a very short number of years. How Eric showed over 2,000 loci for common disease were identified. And you'll see that most of them, we've identified the locus, but we don't know what they work. We don't know all the genes and the RNAs and everything that's involved there. And it's really important to go and understand that before we can just apply the information that comes from the genetic testing. But in this case, I'm going to show you how one of these loci for colon cancer has to do with immunity, not just a cancer cell. And finally, I'll talk about looking at cancer mutations in tumors, patients with advanced disease, and see how that is being used, at least in a clinical trial setting, to select patients' therapies. So to start with the first part, I'm going to start a project with Genome Canada funded our lab, a collaboration between McGill and Genome Quebec when I was still there, and the Ontario groups of Steve Gallinger. We were looking for predictors of colon cancer. This is one of the first genome-wide scans that was done after the HapMap project, where we could take 600,000 SNPs, polymorphisms, and look at large number of cases, large number of controls, and actually see which of those variants actually are different between cases and controls. And what we were looking for is the ability to say someone's at higher risk, and if you know you're at higher risk, well, maybe you should do colonoscopies and colon cancer screening earlier or more frequently, because we know that if you pick up colon cancers at an early stage, the cure rates are pretty high, but if you pick it up in stage 3 or stage 4, it's pretty low. So its ability to actually identify who's at risk would actually bring a modification, which is more intensive screening. The first polymorphism we identified was actually very sobering, actually very disappointing, because the difference between cases and controls was only 17%. So for those who don't do this on a regular basis, all these loci Eric showed have a plot like this in the paper in Nature Genetics. There's an axis which is at 1, and if you see at that a dot was over the 1, that means that there's no difference between cases and controls. Here, you see it's about at 1.17. That means a 17% increase in risk. It's actually 34% increase in risk if you have 2 of the rare mutations, because this is a common polymorphism, common genetic variation. Actually, it's 50% of the allele frequency. So in this room, a quarter of people have none of this risk variance. Half of the people have 1. And 1 quarter have 2. So the risk is 1, 1.17, or 1.34. Not a lot of information, because if your lifetime risk is 6%, then your doctor would say, well, your lifetime risk went to 7%. By itself was sobering, because the marker, the information by this single test doesn't give a lot of information. You get a lot more information from knowing that how much diet you take, for example, how much fiber is in your diet, than you have from this specific test. But as the loci, as that was the first genome scan in 2007. I think there's 22 loci have been identified for colon cancer. Their odds ratios are all low, so individually don't predict much information that's clinically useful for a patient. A few cases we actually know the gene for cancer loci. So there's for example, nicotine receptor is actually the polymorphisms there correlate with lung cancer. But for most of them, we actually still don't know what they do. So I'm going to show in my second part of my talk where it's actually interesting to take one of these validated biomarkers and actually go and say, what is the link between the biomarker and the disease? In terms of risk prediction, we're starting to build models when you have multiple genetic risk, is the information from having all this information together as opposed to a single marker more useful. So this plot basically shows you that the more risk variance you inherit from your parents, the more your risk increase for colon cancer. And because we're all carriers, we haven't seen anyone who carries none and nobody who carries them all. Basically, these are all common variants. Having 9 risk alleles for these are the first 10 genetic markers for colon cancer were identified. So you can have at each of these loci from mom and from dad, you could have inherited 0, 1, or 2. So the most you could have is 20 risk alleles for these 10 markers, and we haven't seen anyone like that, and we haven't seen anyone with 0. But if you have more like 14 or 15 risk alleles, your risk is about 2.5-fold compared to the general population, which the most frequent group, which is 9 alleles. And if you only have 4 or 5, it's around 0.4. So there's a 12-fold increase in risk between having multiple alleles versus having a few. But these subgroups are very rare. They represent less than 1% of the population. Again, not very useful for when most of patients with colon cancer are around 8, 9, 10 alleles. Things become more interesting when you start modeling not just the risk factors, but the age, the sex, and family history, because even family history gives more some additional information than doing genetic markers, because the genetic markers are not testing all the risk variants. So this is a paper of 40,000 individuals, which were groups from Europe, UK, other groups in Europe and Canada together, where if you have more than 14 alleles, you're male, and you have a family history, your risk is around 45% of having colon cancer. If you were aged for the 12 and 13 alleles, which represents about 10% of the population, if you were 50 and you're male and you had 12 or 13 alleles in a family history, your risk of having colon cancer in the next decade is around 15%, and some of them are 20%. So this is the type of population we'd like to be able to screen more effectively, start earlier and maybe more intensively, versus maybe some other groups might need less screening. So these are the types of things which are going to be which we're going to try to elucidate through population research studies. In terms of identifying what is going on locus, again, we use genomic technologies very often. For this one, we decided to resequence all these loci, and I'm only going to focus on one of them, on chromosome 11, which actually had a risk increase of around 14%. And it's just to show an example, but each of those loci need to be studied to understand the biology before we can all use this information. So we used different genomic technologies to be able to resequence the variants, the chromosomal regions in large number of cases and controls. And then we tested all these variants in 1,000 cases with colon cancer and 1,000 controls to see what's going on. Is this the pointer here? Yeah. So the little red dot here is the genetic marker that brought us to that. That was one part of our genome-wide scan. And here we're right under about 4 genes, which are, Eric said there's a lot of genes which we code for protein. In fact, only 1/3 of them do we actually know what they do. These are actually our predicted proteins because they actually have a structure which looks like a protein, but they haven't been characterized yet. And that's what this C11, ORF 92, 93, 53 means. So we started to do the study of these genes because they're right over the disease region. To make this simple, I'm going to go this way. So what do? Well, we actually get tumors from patients with colon cancer, and we get tumors of patients which have the 3 genotypes, AA, which is protection, CC, which is risk, and AC, which are heterozygous when you have 2. And we look at the tumor, the normal tissues. Here, BA means benign adjacent tissues. This is normal tissue. And then we'll look at the tumor tissue, and we see, are there differences in any of these genes between whether you're risk or protective alleles? And in fact, you can see there's a correlation. People with protection produce more of these proteins. Again, we don't know what those proteins do, but these putative proteins, you produce more if you have the protection for both these proteins. So that makes them good candidates for further study. And now when you look at the tumor, you see again the same thing, and particularly here, the people which with the CC, the risk genotype, actually make less of these proteins, ORF 92, 93. So this, with the availability of tumor tissue and normal tissue, allowed us to make see these expression changes which correlate with risk. Now, the 2 proteins are actually regulated together. So there's about 1,000 pairs of genes in a genome where you actually have genes which are oriented away from each other, and the regulatory region, what controls these genes, is actually right in the middle, and since they're on opposite strand, they actually often go up together and go down together, which kind of explains the results we've seen so far. And so there's in vitro tests which allow us to, take the set of polymorphisms associated with risk, and they're in blue, and the set of polymorphism associated with, protection, which is blue, and risk, which is red, actually put them in an in vitro model in cells, actually show that there's more of a reporter gene if you have protection, which correlates again with higher expression going with higher expression going with protection in an in vitro model. What's more interesting is when we start looking in tissues. So most of the colon cancer genes, or any cancer genes that have been identified so far, are active in the tumor cells, in the cells that become tumors. And this is the first one, well, there's a few rare examples, but this first of the common alleles to be shown that it's in the microenvironment. It's not the cancer cells that we seem to be affected by the cancer gene that was inherited. It was the microenvironment. So for those who used to look at pathology here, in brown we have tumor tissue. In red, it's the marker called COCA1. We call it colon cancer associated 1. That's COCA1. And you can see that here, away from the tumor, you can see red. That's not in the tumor cells, but it's in the microenvironment. And here, as you get closer to the tumor, my pointer doesn't work well, you see more red. And in the tumor, you see less because it's mostly tumor cells, very little microenvironment inside that tumor. But if you compare the top panel to the bottom panel, you realize that the microenvironment of someone which had protective alleles, they have a lot more red in all panels than they do if they had the risk genotype. So again, it's not just the RNA. Now we have the protein in the colon tissue, which is overexpressed, this new protein, overexpressed if you have the protective genotype. COLCA2, that's the second gene. That's the other one, ORF93. Again, colon cancer associated 2. And we see again the same thing. We see the expression in the microenvironment. We see a little bit of expression. We see a little bit of pink in tumor cells. So it's not just the microenvironment cells for this one. We see these are tumors, the cells that will become tumor. We see a little bit of pink. And then, and again, you see this difference, a lot more pink and red in the top panels if you have protection versus risk.